Well, that's
How are you?
good. Okay, welcome everyone to another Journal Club. Today we have Dannies Mashabela. He is a first-year Ph.D. candidate at the University of Johannesburg. His supervisor is Dr. Mhlongo. So he completed his master's Cum Laude at the same institute, the UJ, on the metabolomics studies based on Rhizosphere metabolite profiling and metabolic analysis of the plant protective effects of beneficial rhizobacteria on wheat plants responding to a stripe rust and aluminium combination stress. That sounds very interesting. So I believe you're going to tell us the story about your masters. So Dannies, I'll hand it over to you. Good luck.
Thank you so much. Thank you for the introduction. So yeah, today I'll be sharing some of the work that we did for my master's project. So this is mostly from a paper that was published in Frontiers. It's entitled Untargeted Metabolite Profiling to Elucidate Rhizosphere and Leaf Metabolome Changes of Wheat Cultivars that were treated with the plant growth promoting rhizobacteria, Paenibacillus alvei, and Bacillus subtilis. At the end of, towards the end of the presentation, at least, I'll also include some of the work that was not published in this paper specifically, but made part of my M.Sc project and was published in another paper that was more or less like a follow-up of what we were able to find from this study. So thank you for the opportunity to be here. I'll just begin with the presentation. So this would be just a basic outline of what I'm going to be talking about or going through the whole publication. So the global population is growing at an alarming rate. It has sparked a high demand for sustainably sourced food, at least. So this made worse by the factors that are associated with climate change and environmental stressors, which leads to reduced crop yields and have so far threatened food security. And so over the years, we've, as scientists, tried to understand how many plants and many crops in these types of climatic conditions that have become prevalent have been trying to actually survive, and how we can be able to maximize their mode of survival. So plants generally employ strategies from their innate immunity to defend against stressors. However, these defense mechanisms can be breached, right? So we know that if there's a disease breakup of any type, some plants can be able to defend themselves against such diseases. Some unfortunately become susceptible over time, and those defense mechanisms that the plant or crop are even initially applied to protect itself can be breached and would lead to such a plant or a crop being susceptible to the disease and lead to biotic and abiotic stresses. So on the figure that you are seeing on the screen now, it shows some of the stresses that plants generally have to deal with pretty much on a daily basis. So sustainably and environmentally friendly methods for crop protection and growth enhancement are thus a great importance to explore, such as the application of plant growth promoting rhizobacteria. So PGPR, these are the plant growth promoting rhizobacteria, are plant beneficial microbes that inhibit the rhizosphere. So this rhizosphere is basically a region of soil between the plant roots and the bulk soil, which is like very rich in a biodiversity of microorganisms, some of which, like the PGPR that we're going to be talking about here today, have been reported to improve plant growth and development, as well as conferring protection or some level of protection to plants against both biotic and abiotic stresses through either resistance or induced systemic resistance. So some of the functions of these plant growth promoting rhizobacteria are outlined in the figure that you see on your right there on figure 3. So these can be either direct mechanisms or indirect mechanisms. So particularly directly contribute towards the growth and development of the plant, and the indirect mechanisms are more or less associated or concerned with conferring protection to the plant itself. So direct mechanisms just if you include phytohormone production, nitrogen fixation, phosphorus mechanisms, which are to assist with defense of the plant, include the production of inhibitory cytorophores. These are cytorophores that help fight against pathogenic invasions. So they also produce antibiotics and somatic enzymes and induce systemic resistance through some of these compounds, as well as plant priming. So the rhizosphere as such functions as the primary ecosystem for biochemical signaling that's linking the PGPR and the associated plant roots or the plant itself. So this region of the soil root interface presents a favorable environment for the product for PGPR accumulation and proliferation through constant supply of nutritional compounds. So these would be compounds that are actually produced by the plant itself that are secreted into the rhizosphere, and then those compounds become favorable to certain types of bacteria. So PGPR then produces and secretes plant beneficial compounds to promote defense signaling and plant growth. However, the complexity and the dynamics of the rhizosphere metabolome and the effects of PGPR on plant metabolome are yet to be fully elucidated, right? So we are going to try and comprehensively characterize and profile the rhizosphere, or that's what we did in this study, so that we can be able to shed some light on the varied mechanisms by which plants communicate with and are impacted by things from their rhizosphere microorganisms. In this metabolite profile seed bioprimed with cultivars to reveal classes of metabolites, creating the much elusive chemical diversity of the rhizosphere. So for this purpose, we employed metabolomics to comprehensively analyze these small molecules. So on your screen, and what you're seeing is the basic workflow of both targeted and untargeted metabolomics workflow. I'm not going to go into much detail with this, but what we are employed for specifically for this study, we follow the untargeted metabolomics workflow to be able to produce the results that I'm going to present to you shortly. So now the aim of our study were to study the were to elucidate metabolite perturbations in the rhizosphere by plant growth promoting rhizobacteria inoculation in the roots of the wheat cultivars. We also aim to determine the metabolic programming in above ground tissue. So these are the changes that occur in the above ground tissue. So we focus specifically on the leaves here that are associated with rhizosphere PGPR treatments. So the objectives were to profile the rhizosphere metabolome of uninoculated versus PGPR inoculated cultivars. So PGPR inoculated cultivars were those cultivars whose seeds were actually bioprimed with PGPR before planting. And then we used metabolomics to investigate the PGPR metabolic reprogramming that were leading to induced systemic resistance. So this is the methodology that we more or less applied. So the figure that you're seeing shows you the step-by-step where the seeds of the wheat cultivars, these were susceptible wheat cultivars and resistant wheat cultivars to stripe rust. So these were bioprimed, that is where they were coated with the bacterial strains, and then they're planted in perforated falcon tubes. And then from there, both the leaves and the rhizosphere were extracted and then were analyzed using LC-MS. So these extracts were then subjected to UHPLC Q12 HDMS analysis to produce the results that I'm going to present to you right now. So we were able to acquire BPI chromatograms. These are base peak intensity chromatograms that are representing clear differences in the qualitative and the quantitative metabolic profiles of the samples, as shown in the black boxes. So these basically indicate the differences in the metabolic profiles of the cultivars. We used the Harib cultivar, which is susceptible to stripe rust. This was those that were treated with Bacillus subtilis, Paenibacillus alvei, and others were left untreated, which were the control samples. So what we are able to see here now is basically based on the presence or absence of certain peaks, or the presence in different intensity of certain peaks, the differences in the metabolic profiles of the rhizosphere, as of the rhizosphere, as well as the leaf metabolomes of the different cultivars that were treated with the different bacterial strains. So now we then applied chemometric analysis to further reveal the underlying structures and patterns or trends that are in the data, basically based on the different treatments. So the PLSDA scores that you see on your screen now, it shows the separations in the data. Here we observed a clear separation between the PGPR-treated and untreated samples as an indication of differences in the rhizosphere metabolic profiles. Right. So we also observed that the different PGPR treatments resulted in varying metabolic profiles, suggesting that different plant beneficial microbes produced and secrete variant classes of metabolites into the rhizosphere. So this is where we looked at the impact that the treatment with Paenibacillus alvei had on the rhizosphere as compared to the treatment with Bacillus subtilis, and we're able to realize that the two different PGPR actually could have different levels of impact or contributions into the rhizosphere. So it's not necessarily a clear-cut case of it's all plant growth promoting rhizobacteria, then they should all probably secrete the same types of compounds or the same amounts in terms of quantities of compounds that are beneficial to the plant. So different, so here we're able to see that different PGPR strains or bacterial strains can produce different metabolites. They can also be able to have different impacts on the plant itself. So in terms of like the classes of compounds they produce, and at what times those classes of compounds they produce are actually secreted into the rhizosphere. So this phenomenon can also be plant specific, so or dictated by the environmental conditions, as has been previously reported that different plants will attract different microbes into the rhizosphere based on their nutritional or defense requirements. So as I just mentioned, so the different types of bacterial strains can produce different classes of metabolites and secrete them into the rhizosphere. So now what we also observe is that different plants can be able to attract different types of bacterial or PGP into the rhizosphere based on their need, actually. So in a case where the plants just requires nutritional supply for it to sustain its growth and development, there are certain types of microorganisms that it can be able to attract using or exuding specified metabolites into the rhizosphere. Or if it's a case where the plant is facing a challenge, either biotic or abiotic stress, it can be able to excrete certain types of metabolites into the rhizosphere to attract a different type of bacteria strain that would be able to provide it with the necessary metabolites for it to be able to induce or assist with plant defense against the biotic and abiotic stresses. So yeah, it was also observed that the metabolic profiles of the rhizosphere change over time as well. So in the figure as well, we're able to, so that's figure 7B and E, we are able to track the metabolic changes of the different cultivars over time. So the changes over time essentially just represent how the bacteria or the rhizosphere itself changes in terms of like metabolic compositions or profiles over time. So that is more or less expected, as with bacteria growing in an environment, they'll keep on secreting different kinds of compounds over time. The same applies with plants, essentially. As plants grow, as plants develop, they'll over time produce different kinds of metabolites. So to determine the classes of metabolites that were present in the rhizosphere itself, so we carried out a global metabolome view of the rhizosphere using molecular network tools. So this approach revealed metabolites such as organic acids, lipids, benefit from the rhizosphere of PGPR-treated and untreated plants at varying quantities. Right. So, for example, so what we're looking at here, what we're looking at here in Figure 8 is that the rhizosphere soils of plants that were treated with Bacillus subtilis. This is represented by the red indication on the pie charts. There's also the key at the bottom right there. So these were more enriched with benzoates, which are considered agriculturally relevant specialized plant metabolites and are important factors in plant microbe interactions. So this is particularly in parasite recognition in cereals. So cereals are including more or less plants that are or crops that are related to what we're working with here with wheat. So it's been found that the benzenoids considered agriculturally relevant specialized metabolites. So in this case, what we are able to deduce from this is plants that were treated with Bacillus subtilis were able to, or the rhizosphere of those plants was more enriched with these benzenoids. And if they are agriculturally relevant specialized metabolites, it means that the treatment then of crops with this PGPR Bacillus subtilis could be a very important step towards providing crops with sustainable growth, development, as well as plant protection. So there are also, they say that also the plant microbes that are actually important in plant microbe interactions. So these metabolites are essential in the recognition of parasites. So it is essential for these compounds to be present in the rhizosphere in a case where if the plant is experiencing a pathogen invasion, for instance, or a soil-borne pathogen in particular, these compounds are then able to alert the plant that there's an invader, and then the plant can then also be able to produce certain compounds that would be able to attract the microorganisms that are essential for its survival in the rhizosphere. So these phytochemicals are essentially ubiquitous in grasses and as general defense metabolites display allelopathic, anti-feeding, and insecticidal and antimicrobial activities. So it is important based on this to understand how great as a contribution this type of bacteria can be to crops. You know, if they are able to, if for instance Bacillus subtilis, if it's able to produce compounds that are allelopathic, that are anti-feedant, insecticidal and antimicrobial, this can be a great step towards the improvement of plant defense against pathogens, or mostly biotic stressors. So organic acids were also detected which are the most common rhizosphere metabolites. So these are the most common because compounds into the rhizosphere. So they have drawn much attention from researchers due to their versatile profile as functional rhizosphere metabolites. So these are very, very important in the rhizosphere. So they can also be produced by microbes as well. So they are strongly associated with solubilization of essential nutrients, the stimulation of microbial growth, and the detoxification of toxic metals. So we did a study as well before this one where we're doing more or less a general profiling of different types of cultivars, wheat cultivars, and we were able to report that the organic compounds, mostly organic acids that are produced by these cultivars are able to help with some of the cultivars that are resistant to aluminum toxicity, in a case that the organic compounds are able to detoxify the aluminum, more or less aluminum polluted environment that they actually grow in. So then they are able to help some of these cultivars to become resistant to aluminum toxicity. So this also just goes to show the importance of organic metals, or organic compounds in the rhizosphere. They can be used as a detoxification mechanism to allow crops to grow in more or less metal polluted or metal enriched environments. So they are also used in chemotaxis. So this is a mechanism where plants use certain types of compounds to attract or call to the rhizosphere favorable microorganisms. So these are organic acids are essentially important because, one, they're able to be produced and secreted by the plant itself, and the microorganisms that are involved in the rhizosphere are able to produce the same organic compounds. So this then makes the rhizosphere itself such an essential part of the whole dynamic of plant microbe interaction, because a lot that goes into it and a lot that happens in that rhizosphere basically determines the survival of both the microorganisms and the plants that are associated. So we're able to also find phenylpropanoids. So now these compounds are largely absent from bacteria. So therefore, we can then be able to assume that they were produced by the plant itself and secreted into the rhizosphere as root exudates. So now the speculation that comes from this is that plants exude these secondary metabolites into the rhizosphere to serve as chemotaxis strategies as well. So like I explained, chemotaxis, the strategies that plants use to actually attract favorable microorganisms into the rhizosphere. So these are used to recruit the bacteria into the rhizosphere, to shape the rhizosphere microbial community and facilitate plant microbe interaction, or favorable plant microbe interaction as well. So on the other side, Paenibacillus alvei treated plants had a higher accumulation of lipids, right? So we are able to see this in figure 8D there. So lipids in various forms such as fatty acids are modulators of plant microbe interactions, and plants have been reported to use fatty acids to modulate the rhizosphere microbial community essentially. So in this case, lipids have also been reported to function as lubricants for root penetration through the soil and help in water uptake from the rhizosphere. This is done by altering the soil's hydrolytic properties. So it was then easier to understand than this, that most of these lipids that were found in the rhizosphere were actually exuded from the plant itself because of the required function. So for the plants to extend their roots, they need some form of lubrication to be able to penetrate through the soil, and some of these lipids can be used there. So altering their soil's hydrolytic properties as well helps with water uptake. So this just also goes to show how diverse the rhizosphere is in terms of like what properties actually occur in that. So the mechanisms that occur in the rhizosphere, either through growth and development for the plant, defense, the recruitment of microorganisms, and just the plant itself anchoring itself into the soil. All of these mechanisms actually occur mostly in the rhizosphere, making this part of the soil very, very essential. So now several studies have also reported fatty acids as inhibitors of pathogen growth, so to reduce the occurrence of crop diseases and improve rhizosphere conditions and plant growth. So these lipids are also essential in not just helping the plant anchor itself and draw nutrients from the soil, but also in plant defense as well. So if they're able to exude these compounds into the rhizosphere, they can be able to protect themselves against the invasion of pathogen or any occurrence of like diseases. So now, based on the metabolic profiles that I explored, we then went on to investigate the effect of the PGPR treatment on the above ground tissues of the treated plants. So similar to the differential profiles that were observed in the rhizosphere. So we saw variations in the metabolic profiles of the leaves based on the different PGPR treatment. So we can see this in figure 9 here, in A and C. So these are the two different cultivars, Harib, which is susceptible to stripe grass, and then Konap, which is resistant to stripe grass. So basically this suggests that the below ground plant microbe interaction can actually influence plant response in distal above ground tissues for varying purposes. So this can either be by triggering the production of metabolites geared towards plant growth and development, or the redirection of certain metabolic activities towards plant defense. So that means if we are using these microorganisms in the rhizosphere to help with plant growth and development, we can apply these microorganisms to produce certain or the required metabolites, that would enhance or trigger the or increased rate of the primary metabolism, for instance. Otherwise, if these are required for plant defense, the same PGPR can also be used to apply it into the rhizosphere to produce the specialized metabolites that are required to enhance the rate of the secondary metabolism in plants, geared to basically like redirect the whole metabolic activities of the plant, not the whole metabolic activities, but a greater majority of the metabolic activities towards plant defense. So further evaluation of the metabolic events in the leaves post-rhizosphere PGPR treatment revealed perturbations associated with the primary and secondary metabolites, as I just indicated now. So it's illustrated in the figures on your screen now. So the one on your left in figure B and in D, apologies, in figure B and in D on the so where PGPR treatment induced changes in the TCA cycle, intermediates and amino acids content of primed plants. This was compared to their untreated counterparts. So considering that the TCA cycle feeds into the amino acid biosynthetic pathway, this observation then suggested that the alteration in the primary metabolism of PGPR primed plants was more or less channeled into defense-related protein biosynthesis and metabolic pathway, which could suggest an early induction of defense response for induced systemic resistance. So this is again based on the need of the plant at the time. So if the plant, all it requires is not experiencing any type of stress, all it requires is just to grow in a favorable environment, to develop according to at an optimal rate. What the treatment with PGPR can do is it can channel the plant to actually focus on its primary metabolism that would then help with growth and development. So further evaluation of the putatively annotated metabolites revealed potential biomarkers for plant response to PGPR treatment and plant priming. This was so where PGPR treatment has impacted the phenolpropaline pathway. Sorry, I hope you can still hear me. My laptop is telling me that my internet connection is unstable. So hopefully I'm still on from your side.
Yeah, we can still hear you.
Okay, awesome. All right. Okay, so a positive correlation. So looking at our data, a positive correlation was observed between PGPR treatment and the accumulation of phenolic compounds in plants. Right. So this is looking at the figure on your left, on figure D, which outlines the biosynthesis of one the amino acids, which are mostly essential for the beginning of the secondary metabolism. And this goes to the figure on your right, in figure B, where then the phenylpropanate biosynthesis pathway is shown. Right. So the alteration in the secondary metabolism of the PGPR-treated cultivars could serve as a primary mechanism in plants to deploy a more robust, stronger, and lasting defense response upon pathogen recognition. So this is compared to untreated plants. So the observed metabolic configuration could also be a gateway to PGPR mediated into systemic resistance. So what we're able to see is that plants that were actually treated with the PGPR were able to facilitate a more efficient production of specialized secondary defense metabolites. They had an improved phenylpropanate biosynthesis pathway as compared to plants that were not bioprimed. So this is essentially indicating that this PGPR can be used to induce systemic resistance in plants, which anticipate any pathogenic invasion or some sort of biotic or abiotic stresses. So based on the information that we were able to acquire from this study, we then put the plant defense capabilities of PGPR to a test in a follow-up study. And we found that when wheat plants that are susceptible to stripe rust, which is a wheat fungus, showed a generous increase in specialized secondary metabolites. So this is as seen on your screen there. So this, the heat map that you see was basically the different treatments of wheat plants that are susceptible to stripe rust. So there were naive ones that were infected with stripe rust without any prior PGPR treatment. There's a control, and then there's a PGPR treated wheat plants that were then infected with stripe rust. This was done over a 3-week period. Right. And what we were able to notice from this is that the PGPR treated plants that were then infected with induced systemic resistance led to a sharp increase in secondary metabolites as compared to initially the control plants, as well as the naive plants, the plants that were not bioprimed but infected with the stripe rust. And this to us essentially showed the great impact that the PGPR actually have on the plant itself and the different conditions. Right. So this was then further evidenced by a more resilient defense against the pathogen, where the PGPR prime plants develop some leaf chlorosis and necrosis at the site of infections. So this was an indication of a defense response from the plant itself, where they basically the leaf chlorosis and necrosis at the site of infection was to reduce the infection rate of the pathogen itself. So this was evidenced also by the development of smaller and fewer spores with reduced proliferation on the leaf surface. And this was a great indication of the activity that was induced in or the defense mechanisms that were induced in the plant itself due to PGPR treatment, because this was then compared with the non-prime plants. Then I have a small picture on your left here. So the A was the prime challenged plants, and the B was the non-prime challenged plants. So this was a great observation, if we can put it that way. So it more or less the phenotype that we observed was more or less supportive of the profiles that we're able to see, and those are indication of the mechanisms that are actually that actually are happening in the rhizosphere itself between the plant and the microbes that are involved in the rhizosphere. So this was a great demonstration of the metabolic changes associated with induced systemic resistance as well against the pathogen in the primed plants. So the whole train of activities was very insightful when we observed from what happens in the rhizosphere itself. This is like below the ground into the metabolic profiles of the plants themselves, all the way to the phenotypic observations of what the impact of these microorganisms can actually be. So in conclusion, the chemical space of the rhizosphere is highly diverse and complex. It's highly diverse and complex and has been difficult to elucidate so far. So this study shed great light into the complexity, number one, of the rhizosphere itself. And other than just the complexity of it, the great potential that the rhizosphere itself holds in terms of facilitating plant microbe interaction under different conditions, and how the actions that occur in the rhizosphere can be very, very beneficial to the plant itself. Based on the strategies we applied, metabolomics and metabolomics associated technologies, so these offer great avenues for deciphering the mechanistic nature of plant microbe interactions at a molecular level. So it was through applying metabolomics studies that we were able to shed light on these mechanisms. And with the growing rate at which this field that we are focused on is growing, I think there would be more and more opportunities to actually apply these technologies to help us understand plant microbe interactions better, diseases better, and to be able to help us apply what we do in the lab models because of the knowledge we're actually gathering and contributing to the scientific fraternity to be able to come up with innovative strategies, particularly in sustainable agriculture, which is a field that I'm more or less focused on. Secretary root exudates to attract PGPR, which in turn produce plant protective and promoting metabolites. So this is the form of chemotaxis that plants actually use to attract favorable types of microorganisms into the rhizosphere. So PGPR seed treatment could reduce disease progression, severity, or even infection of treated plants while promoting growth and development. So we were able to see how PGPR primed plants are actually able to mount a more robust and a faster defense mechanism as compared to untreated plants against the pathogenic attack. So in this case, going forward, we'd be able to start working at least on mechanisms or strategies that can enable us to bring this concept of PGPR to like a more applicable stage. So to better distinguish between plant microbe produced metabolites, so in the rhizosphere, isotope labeling can be employed. So this is some of the questions that had come up in the line of the study is, okay, if we were able to see all these metabolites that in the rhizosphere, right? How can we be positive that some of these metabolites are actually produced by the bacteria and some of these could be from the plant, and some form of study design that could involve isotope labeling and some other mechanisms can be used to actually be able to track the metabolic, the origins of some of these metabolites. And if we're able to do that, we can then be certain that certain metabolites can either only be produced by the plant or the bacteria. And in that case, we'd be able to then find a way to do what we can call metabolite engineering. So where we can be able to see that if these metabolites are only produced by the bacteria, and maybe not the plant, but they are essential for the plant, we can then induce the production of that those types of metabolites from the bacteria and then be able to like exogenously apply them actually to the rhizosphere or to the plant to the benefit of the plant. So at the moment, we are able to see the profile of the rhizosphere itself, but we are not completely certain to which degree some of the metabolites are produced by the microbes and to which degree some of the metabolites are produced by the plant. So if we're able to like get to a point where we're able to discern that, then we can be able to do a better job at engineering the rhizosphere better for the benefit of the plants or the crops. So that is my presentation for today. I thank you very much for your time.
Thank you so much, Dannies. That was a very good presentation. So I'm going to open the floor up to the audience. If there's any questions, you can either post it in the chat box here, or you can raise your hand and you ask our presenter directly. So, Dannies, I'm just going to ask you, can you go back to your first slide?
My first slide.
Yes. Oh, wait, stay there. Go back. The last slide.
Okay. Yeah?
Your last slide. You had your email.
Slide.
Yeah, you had your email address there.
Oh, yeah.
There we go. Yeah. So that is great. I always encourage people to contact the presenter if they're interested in their studies or getting involved in the studies. So I see you have all your details there. So that is great. So please do contact the presenter if you want to get involved in the research and find out more about the research, or even speak to the supervisors involved here. So I believe your supervisor is Dr. Mhlongo.
Yes, Dr. Mhlongo.
I'm sure he has a question for you. Dr. Mhlongo, do you have a question for your student?
Greetings to the audience and everyone. I don't have one at the moment, but if I have one, I'll ask.
Okay, I see Luke has a question. Go ahead, Luke. You can unmute.
Yeah, and thanks a lot for the presentation. As we find out, I think here maybe join the.
Oh, you're breaking up, Luke. Luke, can you try again?
Is it a biostimulant that was added? Whatever was manipulated, is it a can you call it a biostimulant that enhances the plant to grow? Yeah, because I missed the introduction, I joined a little late. Yeah. And also maybe follow up there, was it, I don't know, because I missed the study, were they added into the seedlings or the plants, and was it once off or at different stages of the plant growth? Yeah, I don't know. Thanks a lot.
Okay. All right. So to answer Luke's question, considered microbial biostimulants. So there's different types of biostimulants, and microbes are actually considered a subsection of biostimulants. So they are biostimulants that we used, and they were applied once off. So they were applied on the seeds. So we did what's called seed biopriming. So what we did is we took the seeds that we're going to use, we coated them with the bacterium, and then this was done obviously before planting. And then once that was done, the seeds were then planted for the rest of the experiment. So this was done only at the beginning. So it wasn't a continuous application of the microbes into the rhizosphere.
Okay.
Okay, no, thanks a lot.
Does that answer your question, Luke?
Yes, yes.
Okay, great.
Okay.
Are there any other questions for our speaker? Dannies, I saw you wrote, or running your methods part, you said you did the extraction of the rhizosphere. How did you do that?
Okay, okay, so let me just maybe get to that slide. Right, so what we did with the extraction of the rhizosphere is we would collect the falcon tubes. So what the falcon tubes that we grew the plants in, they were perforated. So we poked holes at the bottom of the falcon tubes. We put sand in there. So we grew the plants in sand. And then this was important because it would make extractions much easier. So after collecting or harvesting the leaves, we left the root material in the sand first. And then we took our extraction solvent. What we do is we'd run the extraction solvent through the soil, and then we'd collect the solution at the bottom of the falcon tube. And we did that with like 100% methanol, just to try and allow us to extract as much from the rhizosphere as possible. So we would force the extraction solvent through the whole falcon tube, that is the sand in it, and then whatever comes at the bottom would collect in a fresh falcon tube. And that's the approach that we took in extracting from the rhizosphere itself. And then otherwise with the leaf, it was basically just the normal extraction and sample preparation that we usually do on any metabolomics study.
Okay. Are there any questions anyone would like to ask? There is a hand up. Bello, would you like to ask a question?
Hi, everyone. Thank you, Dannies, for the beautiful presentation. I don't know if you mentioned this already, but I wanted to ask why seed biopriming instead of inoculating the plants with the bacteria that's already in Luya broth or any other broth, for instance, I mean. And follow-up question is there a significant difference with the two protocols of priming?
Okay. So for us, I think with seed priming, it was more of like having the seeds as they germinate.
Can you start again? Sorry,
already,
Dannies. You were breaking up in the beginning. Oh, the connection is terrible.
Coated with the bacterial strains, so the seeds as we can get by biopriming them, and also go there. So I was saying is not good. Okay, so I was trying to say it's as close to the seeds as we can get the bacterial strains or the PGPR, such that upon germination, the sprouting plants are already in contact with the bacteria themselves, and the interaction takes place from the get-go. So in a case where you may opt to actually apply the bacteria onto the soil itself instead of like seed biopriming, that's also an applicable route. But you can use either one. Because I remember Dr. Mhlongo actually did the other one in his own PhD, where he would inject the formulation of the PGPR into the soil. But yeah, we figured PGPR biopriming or seed biopriming was as close to the seed as we can get as far as applying the PGPR is concerned.
Does that answer your question, Bella?
Thank you. Yes, I'm sorted.
Okay.
Thank you.
Great, thank you. Luke, you have another question.
Yes, just a small follow-up question, just of interest, because the topic is quite interesting. Now, you looked at the plant metabolites in this part of the plant, but what other issue? Because you can look at the changes could be related to physiology of the plant. Now, you haven't gone into those details? Maybe we need, the physiology of the plant. How does it behave? That could be also interesting. That's I'm just thinking now myself. Because if as you say towards the end, what is the real application of the data to use it? I don't know whether looking at the plant physiology could one way. I don't know. Yeah, I'm just thinking now. Yeah.
Just thinking out loud.
Okay. And I do agree with him on that as well. So looking at the physiology of the plant could be the next step. So here we were more or less concerned with the metabolic profiles and the supposed changes in the metabolic profiles of the plants themselves. We're studying at the rhizosphere initially, and basically how changes in that place can affect the metabolic profiles of the plant itself. But looking into the physiology of the plant as well, that would be a great step forward. But yeah, we're more or less concerned with the metabolic profiles of metabolic perturbations of plants concerning interaction or plant microbe interactions concerning PGPR actually.
I see your supervisor has a question. Dr. Mhlongo.
Yes, I don't have a question per se, but I want to respond to what Luke is asking, actually.
Okay.
The PGPR we received from the University of Pretoria under Professor Laboskachny. He's a plant pathologist, but he has worked in these PGPR. So the choice of us choosing those PGPR is because from his studies, which I think we can link the manuscript if Luke, you are interested in them. But they've had investigated the impact, the physiological impact, which means that we are looking at the plant physiology in terms of appearance on what these PGPR do to those plants. And they discovered that they promote what we call root elongation, root bulkiness, and also they also promote plant growth, which means that when you compare to controls and everything like that, you find that the growth of the plants that are treated are a bit higher. So for this study, we then opt to see what is happening at a molecular level. So I hope that answers your question to say this was sort of a follow-up study where we got these bacteria that were already studied on the impact. I think some of them, they even studied them for nitrogen fixation and phosphorus solubilization and everything. So there were other studies such as your siderols secretion and everything. Then he shared this PGPR with us, and all we wanted to see is that what do they do at a molecular level?
Okay.
Thank you.
I don't know if Luke is still here.
Yeah, no, thanks. Very interesting study. Thanks a lot.
Okay, thank you.
Danny, just go to your first slide and everyone can see your publication.
All right.
Bello, I see your hand is still up. Do you have a follow-up question?
Sorry, no, I don't. Thank you.
Oh, it's just a hanging hand. So there we go. If anyone wants to read further on the details of the study, it has been published. So please do go and search it and download it and read it. So there's some interest in it. So if there are no more questions, I think we can end at this point. So thank you so much, Dannies, for a great presentation and a very interesting topic.
Thank you so much for the opportunity to come and share what I've been working on.
You are welcome. I hope you progress with your PhD. Are you doing the same sort of research as this or anything divergent?
It's not that divergent, but we're incorporating other strategies and other plans on how to expand the project itself. So we're incorporating other types of biostimulants into the study and other analytical strategies to get more or less a broader picture of like what happens to these plants under different conditions in plant microbe interactions. So we'll also be incorporating some transcriptomic studies as well to try and link them to the metabolomics data that we'll be able to generate from the project going forward. Yeah.
Okay, so we're definitely going to have you on again in the near future, telling us about your follow-up research. Okay. Thanks so much.
I'll be looking forward to it.
We'll find you.